All right, ladies and gentlemen, we are going to go ahead and get started. Thank you, everyone, for coming to Plant Breeding Circle today. We have Mandy Yen with us to present, and she is a PhD student in the Horticultural Sciences Department under Dr. Byrne doing, well, rose breeding. So, Mandy, give her a hand.
Good afternoon, everyone. So my name is actually Muquing, but everyone used to call me Mandy, so I'll just go with Mandy. Today we're going to talk about map construction and black spot resistance and TCL detection in diploid rose. So rose as a crop, as a horticultural crop, is very different from the crop most of you dealing with. Actually, for our crop, it's a highly heterozygous line. And all the populations we're talking about today, they are all F1 populations. First, a little bit background about the light. It's not showing on the screen. I already changed the slide.
That figures.
Yeah.
Never so easy. Why be easy? Give me a second.
Oh, here we go.
Okay.
So, continue the talk. A little bit background of the roses and black spot disease. So black spot is one of the most severe, serious diseases on roses, especially for garden roses. So actually, in another disease called powdery mildew, it's a very severe disease for roses in greenhouses. So now today we're talking about the black spot on the garden roses. So people usually now use agrochemical spray to control the disease. However, we know chemical can cause several, issues on human health, and it may contaminate the environment. So in order to reduce the contamination and also help us to maintain our health, we'd like to introduce the genetic resistance to obtain the final result to control the disease. And based on our experience, usually the excellent resistance sources, they are from the diploid and wild rose species. The causal agent of black spot, actually black spot is a fungus. The causal agent is called diplocarbon rosea. It's not ascomycete. So far, 11 races of diplocarbon rosea have been observed and described by some groups. And in roses, we have both vertical resistance and horizontal resistance mechanisms in our roses. So just to be clear now, just for maybe some of you don't really understand what is vertical resistance and horizontal resistance. So first, about vertical resistance, it's also called complete resistant. So it's the gene for gene reaction, which means in your host you have the resistance gene, and if in your pathogen you have the virulence gene, then it will. In your pathogen you have the avirulence gene, then your host will recognize those and it will show the complete resistance. If you have this resistance mechanism, the vertical resistance, then your host is completely resistant to the disease, which means it prevents the whole infection. The other one, and in roses, so far 3 major RDR, the resistance gene to diplocarbon rosea, have been identified. They are RDR1, RDR2, and RDR3. So they are corresponding to different races of black spot. The other one is horizontal resistance. It's also called quantitative resistance, or it's also called partial resistance. So based on the name, we can tell it doesn't show complete resistance to the disease. Instead, it's more reliable and show broad spectrum against the several races of this pathogen. So we think by pyramiding many resistance genes, it will give us stronger power to show the resistance against the pathogen. And our lab is mainly focusing on the horizontal resistance. So we measure the 2 parameters, LAS, which is the percentage of leaf area covered with symptoms or lesions, and we use percentage as our scoring scale. And the other one is LL, so lesion length. We measure the largest lesion, we measure the diameter of the largest lesion on the leaf. Further, why I want to talk about the genetic maps of diploid roses, because our ultimate goal is to detect QTL for black spot. And we all know that in order to detect for the QTL, we need to have the genetic map and the phenotypic data. So before I get started constructing my own genetic maps, I'd like to give you a review about the current study until now, people, what they've been doing. So in the past several decades, we can see, I summarized some major papers work here. So people mainly use these kind of PCR-based molecular markers to map, to create the genetic maps. And some of you may wonder why, for example, in this paper, why they have two numbers in a map. Because for us, I mentioned for our material, they are highly heterozygous. So in order to create one integrated map, we usually do, it's called a pseudo test cross. So we create one paternal map and one maternal map for female and male parents separately. So the number on the left is actually it means the map for the female parent, and the number on the right is the number for the male parent. So yeah, just to give you a general idea, so to summarize, we can see along the time, people increase the number of markers to create the map, and also increase the map length a little bit, because with more marker, you will have a better coverage of your genome. And also the mean distance got decreased. However, so far with so many genetic maps being published for roses, no matter it's for diploid or tetraploid, still no QTL for black spot has been reported, and this is our interest. Before we start to map for the QTL, first step is we need to create a consensus map for our germplasm. All the mapping materials we're using, they're all diploid. And because due to the interspecific nature of roses and it's highly heterozygous, the fertility and the germination can vary among different materials. So in general, our population sizes are pretty small. We tried our best to pick up the three pretty big populations, and also other reasons including because we're interested in these parents. Usually we know in order to create the mapping population, we want to make crosses between one highly resistant line and one susceptible line to create the segregating population. So here I'd like to explain our population to you. We chose these three populations to create our consensus map for our own germplasm. So J14-3, it's our breeding line. It's highly resistant to the black spot. And Old Blush, it's here. It's actually a commercial cultivar, and it's been out there for quite a while. And it has medium resistance to the black spot. And we made crosses between the resistant lines, and LC, VS, and Red Fairy. They are Little Chief, Vineyard Song, and Red Fairy. These three are commercial cultivars, and they have very good horticultural traits, like they're ever blooming, and we like their flowers, we like the flowers color, and etc. However, they have very high susceptibility to the disease. So we created the crosses between two different materials, and that's our mapping populations. As I mentioned, because our population sizes are so small, so it's challenging to do the single family mapping, QTL mapping for our population. So finally, we're gonna do the pedigree-based QTL analysis, and I'll mention that later. So you may feel confused right now why I'm showing you all 15 diploid mapping populations here, because we first started creating the consensus map using the 3 populations out of all totally 15 populations. But later, we're gonna do the pedigree-based analysis combining all 15 diploid populations. So in my project, all my 15 diploid populations, they are related via this pedigree. And on the top, they are all founder parents, which means the parents of these founder parents, they are unknown. So we have no clue about those parents. And Rosa vitriana basiciolis and Old Blush, we believe they are two resistance sources for our germplasm. The Rosa vitriana basiciolis, it has highly resistance for the black spot, and the Old Blush, it has medium resistance to the black spot. And down in the pedigree here are our 15 populations, and I highlighted the 5, 1, 2, 3, 4, 5. Yeah, I couldn't find them exactly, but anyways, there are 5 black spot resistant breeding lines used in our research, and all these 5 highly resistant breeding lines, they are derived from Rosa vitriana basiciolis, which means we believe if we detect any QTL or any resistance genes in these mapping populations, then the resistance sources may come from Rosa vitriana finally, or Old Blush. And first, let's talk about the phenotyping part. So we use the detached leaf assay, so it's a lab-based assay. Basically, we went to the greenhouse and collected the expanded leaves, and then we sprayed them with single conidia race 8 of black spot. Why we use single conidia inoculation? Because we've tested our germplasm with this race 8, and we didn't see any vertical resistance response in our germplasm. So we decided to use this single race to mimic the horizontal resistance mechanism in our germplasm. So after about 2 weeks, after the inoculation, we collected the data. The whole experiment was repeated 3 to 4 times, depending on the material. And based on whether acervali, so this is the acervali, it's the fruiting body, so reproduction part of the pathogen. So based on whether the acervali was present or not, we separated our materials into whether it's susceptible or partial resistance or complete resistance. And further, based on the lesion reading and the lesion length measurement, whether it's significantly higher or significantly lower, we categorized our materials into susceptible or partial resistance. And we measure the 2 parameters to indicate the disease severity. One is lesion length. So we measure the biggest lesion's diameter on the leaf as the lesion length, LL. And LAS is the lesion area covered by lesion. And the scaling scores are here, with 0 means no lesion was observed, and 1 means 10% of the leaf area was covered by lesion, and etc. Okay, and talking about the genotyping part. So people now are using SNP quite often, maybe sometimes some markers that are PCR based, but anyways, SNP marker is becoming more and more popular. So we use the GBS, genotyping by sequencing technology, to generate thousands of SNP markers, and all the detailed steps are in this flowchart. I'm not going to talk too much about that. But anyways, after we got it's the FASTQ file. Basically, it's the sequencing file. It's the sequencing data of our genomic DNA. And after that, we use the CLC Genomic Workbench. It's a software, and to analyze our data. And one thing I want to mention here is, in order to call the SNP marker, so it's the variants, you need to have the reference genome. However, for us, we don't have Rose reference genome available yet. So we use strawberry as our reference genome. And so now strawberry and roses, they are in the same subfamily, so they are very close to each other. And the synchrony between those 2 species, they were indicated before. So we think using strawberry as our reference genome is a pretty good guide for our study. So, and then we use the strawberry as our reference genome to call the SNPs, and then we use the joint map to analyze our type of data and to generate the maps. And also, since nobody ever used so many SNP markers to construct the consensus map or genetic map for diploid roses, and we are the first one doing this work, so we'd like to include some anchor SSR markers as our quality control markers. So previously there was another consensus map generated for diploid rows. And they use the many bridge SSR markers or anchor SSR markers to integrate at least 5 or 6 population or four populations to create the consensus map. And yeah, by including these anchor SSR markers, they will tell us the quality of our map, and also we will be able to assign the pseudochromosome or linkage groups for our materials. So yeah, basically we use the labeled SSR primers, so they are labeled with fluorescence markers, and then we use the ABI machine to call the peaks of the DNA, and then we transform those peaks into alleles reading. Yeah, and we use the joint map 4.1 to create the map, and as well as graphical genotyping. So we could only use joint map because only joint map can handle our type of data. For us, if we're talking about diploid materials, up to 4 different alleles are segregating because our materials, they're highly heterozygous, and we're talking about F1 population. So, and by using graphical genotyping, you can visualize the double recombination events. So by examining these, we may find some markers that might be in the wrong place, and or like for one individual, maybe there are too many double recombination events going on, then it might be selves, you know. So we might exclude those individuals. And in this table, I summarized the population size and the SNP markers number for our first 3 populations. So we started with around 70 to 90 individuals for each population, and during the mapping construction, we realized some individuals, they may show, you know, the alien allele, so they may be wrong process, or so many double recombination, or, you know, beyond your expected events are happening. Then we excluded those maybe self individuals, and then we were down to 69 and 83 and 82 individuals for each population. So when we started the mapping, we had about 4,000 to 5,000 SNP markers. They show polymorphism. However, during the whole, like, you need to code your marker, your genotype to the software's formatting, and also many markers, they showed identical segregation pattern, and you don't want to include all those. And also many markers, they may just you know, inflate your map length, or they just don't fit in your map. So I excluded about 3,000 to 4,000 markers. And then we ended up with 1,400 to 1,500 markers for each individual map. And for the SSR, we've tested around 15 to 20 SSR markers, and we were able to fit 14, 13, and 18 SSR markers for each population. And this just to give you a general idea. This is the individual map for one of our populations, and all 7 linkage groups are here because based on the knowledge that roses have 7 chromosomes per haploid. So we know we need to create 7 linkage groups, and we named our linkage groups based on those anchor SSR markers. In this table, it summarizes the results of our individual maps and also the integrated map, the consensus map. So ICD means integrated consensus map for diploid rows. So basically, we combine all 3 individual maps into one consensus map. And overall, we increased the map length to 892 because we had so many markers, and we increased the coverage of the genome.
Mandy, quick question.
Mm-hmm.
You said, were your parents, I know Rose is highly outcrossed, right?
Yeah.
Okay, so how did you handle the different alleles? Because each parent would have had most likely 2 alleles at every SNP.
Mm-hmm.
Did you have many cases where you couldn't discern between which parent contributed because they might have been identical in states? I mean, was that a common issue?
Actually, well, it depends, because for some markers you have A by A/C, but for some you may have C A/T by G/C.
So how does the program handle A crossed A by C if you end up with the A allele?
You mean in the progeny?
Yeah.
Ended up with A allele? Okay. Well, it's the formatting of this software, like A by AC. Actually, for your female parent, it's homozygous, and for your male parent, it's heterozygous, but they share the same allele. So the program will recode this into LL crossed with LM. So with LL, it's actually for your female parent, it's homozygous A, right? And then in your progeny, if the progeny genotype is A, then it's double L. So yeah, we just manually recoded all the alleles. Yeah, for some genotyping, if we couldn't find the corresponding coding, we just didn't use those markers, because we already have enough markers. So yeah. Sorry about that. Yeah, and this picture shows you how our integrated map. This is one of the linkage groups. I'm showing you the linkage group 2. And this shows you, this in the middle is our consensus map. And all 3 individual maps, they are on both sides. And you can see the synchrony and the consistency across all 3 populations, which indicates our map, they have high quality. And also more mapping information they're shown in this table. So overall, if we look at our consensus map, we fitted 3,527 markers across all 7 linkage groups. However, as you may realize, we have many markers they are co-segregating, and then we try to bind those markers into bin markers and count our bin marker number, and then we have 820 bin markers on the consensus map. So it's a very high density map. Now here, to refresh your memory a little bit, I told you that we used the strawberry as our reference genome. So all our SNP markers, they were called and named based on the physical position of the strawberry genome. So when we grouped and mapped our SNP markers together with the SSR marker, if you look at the linkage group 1 of Old Blush by fairy population, this is one SSR marker, and this is another one, and this is another one. So in this linkage group, we fit about four SSR markers, and they were reported on linkage group 1 of roses. And you may see like all the SNP markers, they are labeled as CHR7. Actually, it means they are from the chromosome 7 of strawberry genome. So when we examine our results, we realize there's a high synchrony between the diploid rosa and the strawberry. And then we went to the previous study. In this paper, they also indicated such synchrony between the 2 species, and we found exactly the same pattern, between us 2 studies. And especially when you examine our data, like this consistency is present across all 3 populations, very high. And if you go back to look at this paper, they only use the limited numbers of markers to indicate the synchrony. However, for us, we use hundreds and thousands of markers to prove the synchrony. So this is pretty exciting. And of course, we'd like to compare our consensus map with the previous one. And we increased the marker number by almost 5 times. And we increased the map length because with so many markers, we have a better coverage of the genome. And also, we decreased the mean distance between markers. It's a high-density marker. However, we weren't able to improve the largest gap between markers. So it might be the ROSA genomics own issue, or it's the centrometric region. And now let's talk about the QTL analysis of our populations. So because no QTL has been reported for black spot, so we like to do some preliminary results. And we found like among all 15 populations, because most of them, their population size is too small, and then we chose the 5 largest populations to try to map for some QTL, just to give us a general idea. And I just used the same strategy to create individual maps for another 2 populations. And also these 2 populations were created by crossing one highly resistant line or one medium resistant line or cultivar with a susceptible commercial cultivar. And these 2 also, they are highly resistant breeding line that are derived from Rosa witriana, Basis-Knowles. And I want to show you the linkage group too. I just randomly picked one just to show you that across all 5 populations, and you can see the consistency of the marker orders, because this software just shows you like all these markers were shared among all 5 different populations. So which also indicate our maps are in good quality. And then we use the, did I miss anything? Oh, yeah. And then we use the map QTL6 to detect for the QTL. So by combining the genotyping data, your allele data, your marker data, and with your phenotyping data, which is our detached leaf assay data. And then we detected for the QTL because we measured the 2 parameters, LAS and LL, so we detected the QTL for these 2 parameters individually. And yeah, we found some QTLs, but at this point, we probably not very convinced by these results, but because they're still preliminary, but this will give us some ideas. Because as I mentioned, our population size is just so small, so the power to detect the accurate QTL or really to apply the molecular markers into the breeding is still questionable. So, but anyways, I will talk about the results I found right now. So the green bar means the LAS trait, and the blue bar means LL trait. And we detected a different QTLs for these 2 traits. Though we use these 2 parameters to indicate the black spot resistance, but still based on the the way you measure those 2, so they may indicate different QTL or different resistance mechanisms. So it's not very surprising that we found different positions for these 2 QTLs. But anyways, yeah, across all 5 populations, we were only able to find QTLs on these 3 populations. And yeah, for the J14-3 crossed with Little Chief, we found the QTL for LAS on linkage group 2. And we found for J14-3 crossed with Vineyard Song, this population, we found the QTL for lesion length on linkage group 2. And also, yeah, we found 1 QTL for each trait in population Old Blush by Raspberry. All these QTLs were detected based on the lot 3, and the lot 3 value was determined based on the permutation test. I ran 1,000 cycles, and I picked the 950th largest lot score as our threshold. So yeah, as I mentioned, finally we're going to do the pedigree-based analysis. So basically the method we're going to use is called Bayesian analysis. So we need to combine all our 15 small populations and do the pedigree-based analysis. And the Bayesian approach is implemented in the software. It's called FlexQTL. So many tree crops actually are using this software, this approach to detect the QTL, because we believe by combining many small populations, that will increase your power to detect the accurate QTL. And of course, we are breeders. We want to really apply what we've learned and apply the marker we identified to the field, and we can do the marker-assisted selection. So finally, we would identify markers associated with resistance to black spot and do the marker assisted selection or breeding. And with that, we'd like to acknowledge all the funding sources and the institutes. And yeah, I'd like to take some questions. Yeah, Dr. Klein.
I have a question, because in the beginning, you mentioned that this horizontal resistance, that your hope is that you're going to be pyramiding many genes in to kind of give you a more broad and durable. But it seems like all of your resistant sources come from Abasis thornless.
Or old blush. Yeah.
But, you know, so, I mean, why would you think that using ones primarily, because old blush is just moderately resistant, you mentioned, but why would you think that you would potentially get many genes, different genetic sources, if you only started with one source?
Okay, so I'd like to dissect that question into small parts. So to start with, actually, we've tested our germplasm, so they show horizontal resistance. You know, based on your phenotype, you have the, you know, the segregating pattern. So we believe there is horizontal mechanism in our germplasm. And also, I believe the next step would be, for us, we started with only one single resistance source, but there are many maybe other resistance sources. So maybe later we could combine those. You know.
So I have to ask a follow-up to that. So how old is Basis thornless? How long has it been around?
That question.
50, 60 years.
Still holding up fine. So why do I need another source of resistance?
Oh, because it doesn't have very good horticultural traits.
Okay.
So yeah.
But you've
you know.
moved that trait into the newer one.
Yeah.
Yeah.
I'm playing devil's advocate for you. Okay.
So let me follow up then. How many genes are involved in that?
I don't know. I mean, based on preliminary results, many genes are involved.
Many genes in that
Yeah.
bunch. So you'd have to integrate a bunch of different regions out of maybe out of an
We really don't
unacceptable.
know.
Yeah.
You don't know.
Actually, you know, linkage group 2 was reported. It contains some resistant genes on linkage group 2, so it might be interesting.
I guess I have 3 questions, but I'll only bother you with 2 for now.
Okay.
I'll ask the third one afterwards. First of all, why did you go with 5 populations instead of one population of maybe 350 individuals?
First, we don't have such a big population.
Oh, it's difficult to create such a large population?
It is.
Okay. And the second question is, what's the lowest centimorgan you can actually detect with the population that's only recombining with 89 individuals? Because I noticed in your map, you're showing like, you're able to detect like a 0.1 difference in centimorgans. And I'm guessing if you figure out the recombination fraction, you might not be able to detect that. I'm guessing it's coming from maybe missing data points, and that's why you're able to get that. And my worry is if you have that many markers in there, it's almost, you're getting a lot of multicollinearity, which might result in inflated R-squares in your QTL analysis.
Yeah, that's possible. But yeah, but like I said, we use the graphical genotyping, and I examined all the recombination events. Like there was, after I screened all those markers, we didn't have any. I don't think I have, you know, maybe some erroneous markers, but not too many that will inflate your map length. And also, if you compare my map length with previous study, because people usually say like PCR-based markers, they're like more reliable compared to SNP, because for SNP marker you can have many missing data. But like if you compare the 2 maps length, they are very close. So I wouldn't say like we inflated them.
And when you do your flex QTL, you'll have way more individuals, right? You'll have hundreds of individuals.
Well,
Yeah.
the census map is about 250 individuals. But yeah, the experiment was really designed for flex QTL analysis, which, you know, when we do all the stuff, they say, well, we only need 40 individuals per population, as long as we have 10 or 15 populations. And that gives us a good consensus of the the germplasm were working as long as they interconnect.
Yeah, and the key factor for us is all our small populations, they are related. So we also did the representative allele calculation to calculate how many progeny we need to perform the calculation to represent the alleles from the founder parent, our resistant sources. So.
I want to point out that in human genetics, they have to use pedigree analysis and Bayesian statistics to follow this sort of thing. So it's not really a new idea.
Yeah, it's not. Yeah, previously
Yeah,
it was mainly used for human animals, and now we're just using it in our
Right.
problem.
And the second thing is, you mentioned the existence of vertical resistance. Has there been, has the fungus developed resistance to those genes, or, do you know much about that?
Oh, actually, yeah, it's a very good question. So first, our fungus is mainly asexual production. So occasionally, very, very low chances you will see the sexual. So the mutation or involvement of the pathogen is very low. But still, we've only identified 3 major vertical genes that they are corresponding to maybe 3 to 4 races. But still, all there's more than 11 races of the black spot. So your
I
disease,
see.
yeah, your
that
resistance
still
can
gives you
easily
plenty of
overcome.
business. Okay, gotcha. Okay, thanks.
Yeah.
So first of all, thanks for the presentation. It's really nice. So how many sort of major QTLs do you think you're seeing now at this point?
If I had to say, there's only maybe 1 or 2 that
Two?
really interested me. Maybe on linkage group too.
So thinking down the line, how will you use that in an applied breeding program, that information?
Now I wouldn't say we can apply our QTL, what we found right now, to apply to our breeding, because it's still ambiguous. And maybe later
In
we shall
terms-
see. Yeah,
but you know they're there though, right?
yeah, but still, if you see how big the QTLs are, they are expanding a very large segment of the chromosome. So
That's not that big. I mean, in terms of the overall genome, those 2 QTLs cover, what, maybe 5 to 10%?
yeah,
At most?
cover, actually they explained, yeah, at least 20% of the total variance.
Of all 7 chromosomes? Those 2 little segments?
Yeah,
No, she's talking about the variance of the.
variance of the phenotypes. Yeah.
No, I'm talking about the segment.
Oh, the length,
QTL
the length
are-
of the QTL. Oh, well, they're here around. But for this one, it's 20 centimorgan. Yeah.
But if you add them together, it's still less than
Yeah.
40 centimorgans
But
under
our
your little map. So it's pretty
at this point, our major concern is because our population size is very small. So
But what?
because at this moment we believe our population size is pretty small, and we're not sure whether the QTLs we've detected they're real or not.
So why not? I mean, I understand you're going to do the Plex QTL thing, but why not take some markers and work with these to see if you can actually do marker-assisted selection for resistance?
Yeah, I mean, sure, we could try that.
That'd be very simple, right?
Yeah, we could verify the markers that are
Then you'd
associated
be
with
down the road in terms of
yeah.
breeding also.
Yeah, we could try.
Easy to do.
We already have one big population, and we leave that population for validation. Any other questions? Yeah.
This was working on diploid rows. If you were going to look at tetraploid, then how would you go about it?
Okay, that's a very good question.
Or how would you take what's diploid and use that information if you were reading the tetraploid?
Actually, okay, the diploid rosa genome and the tetraploid rosa genome, they also have some translocation events going on. So they're not totally, consistent. So, and also, well, right now, people that they've been doing the tetraploid study, they basically use the diploid strategy. You know, just treat your tetraploid as diploid to analyze the data.
Okay.
So, yeah. But, yeah, it's challenging.
Any other questions? All righty. Well, thank you very much, Mandy. Give her a hand, please. All right. Thanks again, everyone, for coming. Real fast, let me figure out who's coming next week. I did have it up, but then my phone went hey wonky. We have Dr. Carlos Avila. He is our tomato breeder down in Westville. He will be coming to speak to us. Thanks so much, everyone.
Good first line.